Today I'm going to talk to you about vaccines. I'm going to add in a little caveat here that my main focus is on antimicrobial resistance in bacteria, but I will see what I can do to carry you through the very rich history and technologies of vaccination. So here's a little overview of what I'll be talking about today. I'm going to give you a little brief introduction about myself, as I'm originally from Wales. I'll then talk about a history of vaccination, sort of how the vaccination process actually works. Then we'll have a very brief break, just so that we can clear our minds before we then go on to talk about vaccine technologies, and then a little bit about vaccine hesitancy and the future steps of vaccination. So first and foremost, os ti'n gallu siarad Cymraeg, croeso. If you can speak Welsh, welcome. I'm originally from South Wales, Pembrokeshire to be precise, and I grew up very close to Narberth. These are the schools that I went to, from sort of Whitland to Preseli, and then I did move up to North Wales for later secondary school. I then went on to do a course on biological sciences in the University of Bristol. So that was my undergraduate degree. I loved it so much that I stayed on to do a master's working on the Big Tick project. So my initial sort of specialism post-undergraduate research was on sort of ticks and tick-borne diseases, and I got quite into the idea of sort of like pathogens and better understanding pathogen dynamics. And it's from there that I went on to join the University of Oxford, and specifically Jesus College, to do my DPhil, which is in the Griffin Lab, which is a lab that actually focuses on social evolution in bacteria. These are my two wonderful supervisors, Professor Ashley Griffin and Dr. Melanie Gould. So it's from here that I do all of my sort of research, but I also like to participate in outreach as much as possible. Right, so on to our actual sort of introduction into vaccines today. Now this is quite a rogue slide, so bear with me. But this is kind of like touching on hesitancy and fearfulness of things that we don't fully understand historically, and also how they can be quite sort of easily debunked when you actually start getting into the details of this. So this is the story of Robert Gibbon Johnson, who lived in America, in Salem, New Jersey, and in sort of say 200 years ago, so early 19th century, people used to think that tomatoes were poisonous. And the story goes, so I learned this story while I was doing my undergraduate degree, learning about sort of technologies in plants. And my lecturer said that there was this guy, and he wanted to prove to everyone that tomatoes weren't actually poisonous, and that it was just sort of like all of this hype around it. And so the reason people thought that tomatoes are poisonous is because they're from the Solanaceae family, which actually can produce a sort of a toxic poison. But in reality, tomatoes obviously weren't poisonous, and the story goes that Robert Gibbon Johnson did a sort of a public display where he ate a basket full of tomatoes, and all of the bystanders thought that he was going to die. So it's almost like watching someone crazy sort of almost commit suicide by eating tomatoes in front of everyone. But in reality, obviously he didn't die. But then if we actually dig further into this story, it's actually a fake law. So it's not true, but it actually stands to present a very interesting story and an idea in science of how misinformation can spread, and people can think that something is super dangerous when it's actually not. And I mean, who doesn't like tomato-based pizzas and sort of like lovely, delicious tomato pasta dishes to know that they're definitely not toxic or poisonous. But that was a little bit off topic. It was just to sort of present the idea of the public and how misled people can be. Now we're going to actually go back on topic a little bit more, and we're going to talk about today specifically, I guess, vaccination against viruses and bacteria. But just give a little bit of context. Here we have a virus, a bacterium, and a eukaryotic cell. So you've probably touched on these a little bit in GCSE and the first year of A level, but essentially a virus is just sort of like a nucleotide sequence coated in protein that invades cells and replicates itself using the cell's machinery. A bacterium can self-replicate, and they are unicellular organisms with no membrane-bound organelles. And of course, a eukaryotic cell is what sort of makes up you and me. And these are cells that contain membrane-bound organelles, and they form multicellular tissues. So as I said, today we're going to be really sort of zoning in on these bad boys over here. Right then. So just to also give a little bit of context, in terms of sizes, viruses are obviously like tiny in comparison to bacteria, which are already tiny relative to eukaryotic cells. So these are very small pathogens that the eye cannot see. Right then, so now going into the history of vaccines. So when you think of, the creation of vaccines, you probably think about Jenner and sort of the whole cowpox versus smallpox thing. I mean, we'll get to that, but prior to sort of that form of vaccination, a thing called variolation has been around for a very, very long time. And this is more an inoculation kind of process where potentially live viruses or pathogens can be transferred to sort of hosts that have never been exposed before, a little bit like this. So you'd get someone with a live infection that may have sort of antibodies within the pustules. Then you take a little bit of that pustule and you inoculate it into a host. And the idea is that because of the presence of antibodies within these pustules, that will be transferred, conferring sort of better protection to the sort of unexposed person. However, this can go wrong, obviously, because you're transferring likely live virus or live pathogens, and they can potentially just sort of spread this potentially fatal infection. So it's not necessarily like the most reliable thing, but it can be very, very effective. And this sort of long predates Jenner. There are some reports saying that this happened sort of thousands of years ago, and if not sort of reliably centuries ago and centuries prior to Jenner's technique. But we must talk about Jenner here because this is a really pivotal moment in vaccinology because of how much safer this method was. So of course, with Jenner's story, it is very much an observation that cowpox provided sort of a very sort of similar infection to smallpox, but a lot less virulent. And milkmaids would likely get cowpox, and they would unlikely be burdened with smallpox, which was a really big issue in the late 18th century, and sort of a potentially fatal disease that left horrible pockmarks on its victims. So Jenner noticed that there was potentially a link here, and he sort of inoculated a young boy with the cowpox virus, and then a little while later tried to inoculate the boy with the smallpox virus, and obviously realized that no infection occurred due to the similarity in the viruses and the similar sort of antibody response. And so yes, there's no actual sort of direct link between the specific cowpox and smallpox viruses that were, the specific cowpox virus strain that this was proved with. But I mean, it's a very, very pivotal step in the development of vaccines, sort of knowing that similar viruses can protect against potentially more deadly viruses. And what I think is really, really cool is that because this was done using cowpox, the word vaccination actually comes from the Latin word vacca for cow. So I thought that was pretty neat. So how do vaccines actually work? So of course, with a mammalian body, we have an adaptive immunity that enables our bodies to detect and learn about different pathogens and sort of non-self cells that should be targeted and stopped. So we have the key features of the adaptive immune system, and then features that either sort of like spring from that, or we've also got our innate immune system of phagocytes, which can sort of detect and engulf cells. So when we get a pathogen that enters the host and it's not supposed to be there, the dendritic cells detect this and express the antigens present on this pathogen. Now, antigens are just essentially sort of virulence factors that are expressed by pathogens that sort of enable certain sort of host attack systems. But the dendritic cells can express these, which triggers a response from the T cells, the helper T cells, because they detect the expression of the antigen. This then leads them to connect with the B cells, which then convert into plasma cells, which create specific antibodies that will then tag on to the invading pathogen. We also get then T killer cells or killer T cells, which also have specific pathogen recognition, which I'll go on to talk about a little bit in the next slide. So what we tend to see as well is engulfing of the pathogen by the phagocytes. The phagocyte then sort of further engulfs the pathogen into little pockets within the cell and digests the pathogen and breaks it down and renders it sort of no longer virulent. We also, I mentioned the killer T cells. They will actually identify infected cells within the body, and they will attach to them and then again release loads of enzymes that then break down and sort of dissolve this cell and render it ineffective. So with the immune system, we've got this sort of very synchronized, specialized system that can detect invading pathogens and develop a very specific, tailored response so that the pathogens can be stopped and rendered ineffective or destroyed. So it's an incredible system that's so sophisticated, and we're so fortunate that our bodies can do it. I mean, not only does the body sort of have this first attack defense, it also then goes on to create a memory of this invasion for any future attacks. And I'll say here, this video on YouTube is fantastic for a thorough sort of overview of these things. And just to go back to summarize, we've got the dendritic cells, which sort of detect and start expressing to alert the immune system. The helper T cells detect then the production of these antigens, and then that triggers a connection with the B cells, which express antibodies. And then the B cells produce plasma cells, which then further express lots of antibodies. The killer T cells recognize any pre-infected cells and destroy them. The phagocytes recognize any of the pathogen that's present within the host and engulfs it and destroys it. And then for long-term storage memory of this response, we have the memory B cells and the memory T cells. Oh my gosh, I've just realised we are sort of, quarter of an hour in, and I haven't even mentioned the C word. Yes, I haven't talked about COVID yet. And that's kind of a little bit deliberate, because I don't want to make this all about COVID. I mean, our life has been COVID-centric for the last 18 months, but that might be a bit miserable. But what I will say is that obviously with any sort of COVID infection, so COVID is a virus, COVID would trigger this kind of sequence of events, and people that have been infected with COVID do ultimately sort of develop these memory B cells and memory T cells, so that future infection may be fought off, but not necessarily always fought off. Right. And one thing I will say here as well is that, so one thing that was happening with COVID is that people sort of look for antibody presence within people that may have had COVID infections. But the thing with antibodies is that they're quite short-lived in the host, so they're not going to stick around forever, because they're only going to be produced if there's sort of an imminent threat from the virus. And so it's actually the memory cells that are most important here to sort of really make sure that the infection is recognized again in the future. However, with viruses, we tend to get mutations, as you've probably come across in the news. You may have found, things about sort of new variants of COVID and the like. And of course, if we get new variants that are so distinct and different from the original infection, then all of this sort of memory recognition is no longer going to work. And that's why we can get variant escape. Fortunately, with the vaccination programs that have been developed, we actually do see very little sort of escape from the vaccination and sort of occasional reinfections of COVID. But that might be down to the fact that the initial infection or the initial challenge from the virus or virus parts wasn't as strong as it could have been. And so this sort of large defense network of memory cells may not have been as good as it could have been. That was a lot of information. So I'm going to give everyone just 30 seconds to a minute just to take a little breath, have a breather, and realise it's a Monday morning, so you might be sort of a little bit bogged down with all of that information, and I will resume in just a second. Right then, I hope that was enough time just to have a little breather and do something different. I think it's so important when you're sort of taking in lots of information just to have a pause and to think about all of the things that I've just discussed. So how are vaccines made? So I've given you a brief overview of sort of the origins of vaccination and how people kind of saw connections between, a similar disease that was sort of less virulent and a sort of a more sort of sinister disease being more virulent, and how similarities between those viruses can actually confer protection against the more sort of sinister disease, as mentioned with cowpox and smallpox. But I mean, back then, you can sort of see the similarities and use the principles, but obviously for every virus, we don't necessarily have a sister virus that's slightly less virulent that can be used to inoculate with or vaccinate with. So it's a case for modern technology to then take forward this sort of these principles and figure out steps in how to protect the world against these emerging pathogens. So we have a very established methodology, such as using live attenuated vaccines. So these are weakened forms of the virus. Key examples here is the MMR vaccine, which is the measles, mumps and rubella vaccine. And this would involve sort of finding a weaker form of the virus or the viruses, and then inserting them into the host via injection. You can also, depending on sort of target sites of the virus, you can have different forms of inoculation or vaccination, where you can have oral forms, nasal forms, and of course injected forms. But MMR would usually be administered using an injection. And the benefits of live attenuated vaccines is that they confer a massive immune response, because it's the body detects a potentially live virus and attacks it. This is, however, a negative thing for immunocompromised individuals, and this can lead to sort of complications. So these forms of vaccines tend to require a healthy immune system. Another form of established methodology, so this is a means of sort of producing vaccines in the past, are subunit vaccines. Now these vaccines may sort of take a small section of the virus and then remove any of like the pathogenic elements. So for example, the genetic sequence within the virus, like the DNA or the RNA, that actually leads to pathogenesis of this virus. And so these can be injected, however, alone. So this very sort of benign form of the virus won't necessarily confer a massive immune response because the body doesn't detect any sort of imminent threat. But it's actually, it was discovered that sort of injecting with this thing called an adjuvant actually sort of alerts the immune system to create a specific immune response against this pathogen. Again, so the host cells will detect the specific antigens expressed on the surface of this subunit vaccine and develop specific protection, sort of antibodies against it. The good thing about these subunit vaccines is that they're a lot less sort of stress on the immune system. So immunocompromised individuals can take them successfully because they're less likely to actually confer a proper attack. But the issue with attenuated vaccines is that because this is sort of a weaker, a much more benign form of the pathogen, it's actually even harder to develop a significant enough immune response to confer long-term protection against this virus. So it might lead to having to carry out further doses of the vaccine sort of further down the line. So, I mean, great for some reasons, but then maybe not so good for other reasons. Now we're moving on to newer methodology, and this is where the COVID-19 pandemic has led to a rapid explosion of sort of revolutionizing vaccine development. So the one I'll talk about here is the nucleotide-based vaccines. So, for example, an insertion of mRNA into a fatty microdroplet that is then administered into the bloodstream. This will then merge with a cell within the host. And since it's an mRNA sequence, as you probably all know from your protein synthesis topic in school, this mRNA sequence will enter the cell and hijack sort of the ribosomes. The ribosomes will then produce, translate this mRNA sequence to create the protein which forms for example, the antigen. Now, specifically to COVID, this example of vaccine would be your Pfizer vaccine. Pfizer has an mRNA-based vaccine that, exactly as I've just described, this sequence actually produces one of the spike proteins on the COVID-19 coat surface. So that's how it confers protection. It seems to be very effective, and it utilizes the host's sort of replication systems, very similar to old vaccines. And the good thing about it is that you can tailor make this mRNA sequence relative to the specific and viral strains. So, for example, if you get new variants and new strains, you can modify this sequence to directly target that. But obviously, with modifications in sequences, we have to go through safety protocols just the same as we would with a newer vaccine. So I'll talk about sort of the whole deployment protocol in just a second. The other type is using a viral vector. So we have viral vector vaccines where we might use a different virus that might be quite benign and sort of not very infective, but we will take that blueprint and insert our sequence of interest. Again, for example, I believe this is with the AstraZeneca vaccine. We've got a benign virus where we've inserted a small segment of the COVID sequence, so for example, the spike protein, and then that will be injected into the host, and the host will then produce loads of this sequence and produce the spike proteins and ignite a very similar immune response as to the one that I mentioned before. So these are very new technologies, and rather than sort of trying to find sort of elements of the virus specifically via like the proteins or weakened form of the viruses, we actually sort of engineer specific, so very virus specific sort of factors that are then inserted into the host. The good thing about these newer technologies or newer methodologies is that they're very quick to create, and they're very specific to strains or to specific viruses. So that's a massive benefit, and I think that this whole pandemic has really sort of accelerated this technology that was already in existence prior to the pandemic, but of course this is an example of the fastest sort of vaccine development deployment process sort of ever really known. So it's just incredible what's happened. Now I've mainly been talking about viruses, obviously, because we've all been sort of thinking about viruses so much recently, but of course you can use vaccines to treat bacterial sort of infections. Now this isn't the sort of thing that you're likely to do for lung infections or I don't know, specific skin infections or whatnot. This is more for the widespread bacterial infections that can be life-threatening, for example, TB, tuberculosis. And so you can have specific vaccines against TB that can confer protection and sort of eradicate that kind of issue in human populations. So following very, very similar thought process to the other forms of vaccination, you can have weakened forms of the bacteria, bacteria-specific antigens that you can alert the body to, and then again, building on this, the mRNA-based and the viral vector-based vaccines. And again, you can use a viral vector for this because it's just the genetic sequence that creates the antigens on the bacteria surface. But these are like roughly the different types of vaccines. I'm sure there's lots of further resources that you can utilize online, but I hope this overview has sort of given you a rough idea of what's out there. Now on to the development of vaccines. So vaccines would usually take at least 10 years to produce. This goes from sort of the initial discovery. So there's tons and tons of different types of potential vaccines. And especially if we're thinking back here to our sort of attenuated vaccines versus the subunit vaccines, these might actually take a bit longer to isolate and to trial in sort of lab-based experiments. And we might actually find that because they may be so benign or non-virulent, they don't actually confer any response within lab experiments. So a lot of these initial vaccines will just be dropped because they're just sort of not found to be effective. Now, the really cool thing about the more modern vaccine technology, so using the viral vectors or the nucleotide-based vaccines, these can be tailor-made very quickly and sort of trialed on a smaller time frame. And because they're engineered by scientists, they're a lot more specific to the direct targets. So it's a more sensitive and refined technology in that case. But historically, that whole step would take at least 2 to 5 years. And obviously for us, it did not take 2 to 5 years, otherwise we'd still be hanging around waiting for vaccines for COVID. You then sort of move on to further preclinical trials, looking again at more sort of lab-based assays, potentially using sort of animal models to detect antibody responses, before sort of narrowing it down to the point where you can reliably get responses and that you go into your phase trial stages. Now this is a much longer process again. So firstly, you start with phase 1. If I inject this vaccine into a human, are they going to die? That is kind of like the ultimate question. But obviously, it's not just are they going to die, it's going to be, will there be an adverse reaction? How severe is this reaction? Can this be overcome? If not, we'll sort of ditch that vaccine. If so, how can we sort of make it reliably safe? So that would be the initial process of the trial. Then we move on to a phase 2 trial where we start then specifically looking for a good immune response to the vaccine. And then this will be narrowed down again. So if you imagine, you know, we've started off with up to 100 potential vaccines, now down to only 5 in this phase 2 trial. This will be a wider group of participants. And then, as I've mentioned, looking specifically for sort of antibodies, successful antibodies. And then once we've got down to that, we might actually sort of narrow down again to vaccines that are sort of triggering reliable antibody responses, and then we'll move into a phase 3 trial where we have sort of maybe even one potential vaccine that's actually been successful, and this now is to prove whether the vaccine can actually prevent disease. So can this stop the virus or bacterium from causing a disease within the host? Are the antibodies sort of strong enough? And this can take anywhere between sort of 2 and 4 years. So again, a very, very long process. Then all of the data has to be collected and all of this has to be reviewed until it can be approved by sort of a panel of specialists that agree that the vaccine is safe and that it can be deployed. And then, and only then, we get into the headache of manufacturing and deployment. So this is where the COVID-19 vaccine process has been, you know, really phenomenal, that people have been able to create so many doses in such a short space of time, because obviously you've got to think about the reliability of these doses. They've got to be 100% sort of similar to each other, like they've got to be identical, and that's got to be replicated sort of over millions and billions of doses. So it's very important to have a very strong manufacturing process within the vaccine development and deployment. But obviously I've just gone over this, and this would be the standard methodology prior to COVID for developing vaccines. But with this sort of global vaccine development and deployment that we've had to undertake to tackle COVID, the future of vaccination might actually be a little bit different and a bit more tailor-made, and may not take such a long time. However, of course, costs may be quite constant. So just to go over, with vaccines, you have a very specific sort of 3 trial phase at least, just to make sure that these are not going to, you know, sort of using a vaccine that's going to lead to the demise of the human race or something as dramatic as that. But the good thing about COVID, even though it was the vaccine was developed and deployed in a very, very short space of time, it did not skip any of these trials. So it underwent all of these phases. Yes, they were massively sped up. So the process was expedited massively by just quicker verification of the results between each phase. And then also, I'm not sure if this was the case with COVID, but you can also run some of the phases concurrently if the preliminary results of the other initial phases are looking good. So this is all extremely relevant in terms of fighting COVID and potentially future pathogens. Touch wood that there aren't many, but yeah, I think it might be inevitable. Going on then, so with a vaccination program, not everyone needs to be vaccinated. It's just the vast majority. I mean, the more the better, because of course it confers more resistance in the population. But the good thing about herd immunity is that if you get a good sort of take up of the vaccine in the vast majority of people, then these vaccinated individuals in blue here will protect the unvaccinated individuals in red. And people that may not have taken up the vaccine, for example, can be sort of immunosuppressed individuals that aren't necessarily healthy enough to take the vaccine. So it's great if the sort of vast majority within the population can take the vaccine and protect these individuals. However, obviously, not everyone sort of decides not to take the vaccine because of specific immunosuppressed health conditions. We also get vaccine hesitancy, people that are afraid of taking a vaccine for fear that it will lead to adverse effects in their health, and maybe even a government conspiracy like how Pfizer can now give you 5G signal, which sadly it can't. I would almost welcome this. I have no signal on my phone ever, and I still have no signal on my phone ever. So I can confirm, at least for me, Pfizer does not lead to a 5G boost. But as outlined by the World Health Organization, vaccine hesitancy is one of the top 10 threats to global health. And this is in 2019, prior to the COVID-19 pandemic. I believe at least, I think, 1.5 million deaths. Wow, there's a lot of millions. I can't remember the specific statistics, but millions of people die each year due to vaccine hesitancy, whereby we've got successful vaccines to fight against diseases and they're not necessarily used to confer protection, and as a result people die needlessly from these infections that we've already learned how to fight off successfully historically. So I'll tell you a little story about vaccine hesitancy. There's a lot of sort of thoughts, well, there's lots of worry and concern about the vaccine for MMR, measles, mumps and rubella, leading to the development of autism in children. Now, the interesting thing about the MMR vaccine is that it's usually administered between, well, at around 15 months in infants, and then a follow-up vaccine at 4 years. Now, interestingly, as outlined by Tim Harford in How to Make the World Add Up, which is a fantastic book for any sort of like number literacy reading, you know, just to think about how statistics sort of rules the world and how we can actually think about numbers better. Tim actually sort of identifies a bit of a link between when MMR vaccines are administered and when autism diagnoses are given. For example, at 15 months, you'll be interacting with nurses a bit more for the first time, and you'll be more lucid as an infant. So any sort of underlying conditions like autism may be picked up on around that time. And so people think that my child was just given the MMR vaccine and now they've got autism. So it does kind of lead to a link, but obviously correlation and causation are different things. You then further get with a potential link in sort of correlation with autism and MMR at 4 years, when you're sort of entering primary school level, where teachers may start to sort of pick up on specific underlying conditions like autism, and leading sort of parents to think that the MMR vaccine again sort of led to autism, which may not be the case. And it's not the case necessarily. I mean, the latest research from Denmark followed loads of children from sort of very young infancy through to young primary school age, looking at the children that were given the MMR vaccine by choice of their parents and the children that weren't, and found no significant difference in the cases of autism in either group, leading them to sort of confer that or infer that the MMR vaccine does not lead to autism. So again, this sort of extreme vaccine hesitancy might be a sort of like one of those stories, going back to my tomato story in the beginning, of how future populations may look back and reflect and think, goodness, how on earth did people ever fear this thing that is a life source? I mean, pizza is a life source for us now. Could you imagine it without tomatoes? That seems crazy. Same as vaccines being sort of looked back upon by future generations as something that is just a given in day-to-day life. And what's also really exciting about vaccines is that because we may start being able to tailor make vaccines specific to certain protein structures, we may start to be able to vaccinate against things like cancer. So within an individual that is suffering from cancer, you might be able to take a sample of the cancerous cells and sequence these things called neoantigens, which are present within cancer cells, and develop a vaccine that will then go in and target that cell. So it's just incredible to think that this technology can actually start leading us to protecting us from things like cancer, which is phenomenal. And maybe one of the benefits, and I mean there aren't many, of the COVID-19 pandemic is that it's massively advanced vaccination technology to sort of give us a much better toolkit for battling things in the future. And there are some success stories with vaccination programs as well. In 1966, the World Health Organization launched the Global Smallpox Eradication Program, and by 1980, the world was declared smallpox free. And this all started with Edward Jenner and his cowpox versus smallpox link, and thinking about means of protecting people against smallpox, which eventually led to the eradication of this virus. So we can tackle these awful diseases with vaccines, and can potentially sort of overcome these problems and protect the human race. So just to give you a little overview what I've talked to you today about, we had a brief introduction from me. We then talked about the history of vaccines, the immunology, sort of the method of how vaccines actually work. We had a very brief interlude, and I hope you took a chance to have a breather. We then talked about vaccine technologies and all of the means of sort of developing vaccines, vaccine hesitancy and why people may be hesitant when using vaccines, and the very hopeful future steps with vaccination. Thank you very much for listening to my talk today.